General reactions of amino acids. The reactions due to carboxyl group. The first reaction, decarboxylation. The amino acids will undergo alpha decarboxylation to form the corresponding alanine. Thus, some important amines are produced from amino acids. For example, histidine gives histamine plus carbon dioxide, tyrosine gives tyramine plus carbon dioxide, tryptophan gives tryptamine plus carbon dioxide, lysine gives cadaverine plus carbon dioxide, glutamic acid gives gamma amino butyric acid plus carbon dioxide. The second reaction is amide formation. The COH group of dicarboxylic acid of amino acids other than alpha carboxyl group can combine with ammonia to form the corresponding amide. For example, aspartic acid plus NH3 gives asparagine. Glutamic acid plus NH3 gives glutamine. These amines are also components of protein structure. The amide group of glutamine serves as the source of nitrogen for nucleic acid synthesis. The next one is reaction due to amino group. The reaction is transamination. The alpha amino group of amino acid can be transferred to alpha keto acid to form corresponding new amino acid and alpha keto acid. This is an important reaction in the body for the interconversion of amino acid and for synthesis of non-essential amino acids. The next one is oxidative deamination. The alpha amino group is removed from the amino acid to form the corresponding keto acid and ammonia. Decarboxylation of amino acids in the body, glutamic acid, the most common amino acid, to undergo oxidative deamination. The next reaction is formation of carbamino compounds. Carbon dioxide adds to the alpha amino group of amino acid to form carbamino compounds. The reaction occurs at alkaline pH and serves as the mechanism for the transport of carbon dioxide from tissues to lungs by hemoglobin. The next reactions due to the side chains. The reaction is transmethylation. The methyl group of methionine after activation may be transferred to an acceptor which becomes methylated. That is methionine plus acceptor gives methylated acceptor plus homocysteine. The next reaction is ester formation by the OH group. The hydroxyl amino acid can form esters with phosphoric acid. In this manner, the serine and threonine residues of proteins are involved in the formation of phosphoproteins. Similarly, these hydroxyl groups can be O-glycosidic bonds with carbohydrate residue to form glycoproteins. The next reaction is reaction of the amide group. The amide groups of glutamine and asparagine can form N-glycosidic bonds with carbohydrate residue to form glycoproteins. The reaction of HH group. Cysteine has a sulfidyl group and it can form a disulfide bond with another cysteine residue. The two cysteine residue can connect to polypeptide chains by the formation of interchain disulfide bonds or links. The dimer formed by two cysteine residues is sometimes called cysteine or dicysteine. The final one is amino acid derivatives of importance. Gamma aminobutyric acid, that is GABA, a derivative of glutamic acid, and dopamine derived from tyrosine or neurotransmitters. Gabapentin, an analog of GABA, can pass blood-brain barrier and can form GABA in brain. Gabapentin is clinically used for relief pain. Next, histamine, that is synthesized from histidine, is the mediator of allergic reactions. Thyroxine from tyrosine is an important thyroid hormone. Cycloserine, a derivative of serine, is an anti-tuberculosis drug. Azathioprine inhibits reactions where amide groups are added and so act as an anti-cancer drug. Histidine residues are important in the buffering capacity of proteins. Ornithine and citrulline are derivatives of arginine and are essential for urea synthesis. Protein analysis. Proteins are polymers of amino acids. 20 different types of amino acids occur naturally in proteins. Proteins differ from each other according to the type, number, and sequence of amino acids that make up the polypeptide backbone. As a result, they have different molecular structures, nutritional attributes, and physicochemical properties. Proteins are important constituents of foods for the number of different reasons. They are a major source of energy as well as containing essential amino acids such as lysine, tryptophan, methionine, leucine, isoleucine, and valine, which are essential to human health but which the body cannot synthesize. Proteins are also the major structural components of many natural foods, often determining their overall structure, example tenderness of meat or fish products. Isolated proteins are often used in foods as ingredients because of their unique functional properties, that is, their ability to provide desirable appearance, structure, or stability. Typically, proteins are used as gelling agents, emulsifiers, foaming agents, and thickeners. Many food proteins are enzymes which are capable of enhancing the rate of certain biological reactions. These reactions can be either a favorable or detrimental effects on the overall properties of foods. Food analysts are interested in knowing the total concentration, type, molecular structures, and functional properties of the proteins in foods. Determination of overall proteins concentration. That is, first one is Kjeldahl method. The Kjeldahl method was developed in 1883 by a brewer called John Kjeldahl. A food is designed with a strong acid. A food is digested with a strong acid so that it releases nitrogen, which can be determined by suitable titration technique. The amount of protein present is then calculated from the nitrogen concentration of the food. The same basic approach is still used today, although a number of improvements have been made to speed up the process and to obtain more accurate measurements. It is usually considered to be the standard method of determining protein concentration. Because of Kjeldahl method does not measure the protein content directly, a conversion factor F is needed to convert the measured nitrogen concentration to a protein concentration. A conversion factor of 6.25, that is equivalent to 0.16 nitrogen per gram of protein, is used for many applications. However, this is only an average value, and each protein has different conversion factor depending on its amino acid composition. The Kjeldahl method can conveniently used divided into 3 steps: digestion, neutralization, and titration. Principle. The first one is digestion. The food samples to be analyzed is weighed into a digestion flask and then digested by heating it in presence of sulfuric acid, an oxidizing agent which digests the food. Anhydrous sodium sulfate to speed up the reaction by raising the boiling point, and a catalyst such as copper, selenium, titanium, or mercury to speed up the reaction. Digestion converts any nitrogen into the food other than that which in its the form of nitrates or nitrites into ammonia and other organic matters to carbon dioxide and water. Ammonia gas is not liberated in an acid solution because as ammonia is in the form of ammonium ion NH4, which binds to the sulfate ion, that is SO4 2- and thus remains in solution. The next step is neutralization. After the digestion has been completed, the digestion flask is connected to a receiving flask by a tube. The solution in the digestion flask is then made alkaline by addition of sodium hydroxide, which converts the ammonium sulfate into ammonia gas. The ammonia gas that is formed is liberated from the solution that moves out of the digestion flask and into the receiving flask, which contains an excess of boric acid, The low pH of the solution in the receiving flask converts the ammonia gas into the ammonia ion and simultaneously converts the boric acid to the borate ion. The final step is titration. The nitrogen content is then estimated by titration of the ammonium borate formed with standard sulfuric and hydrochloric acid, using a suitable indicator to determine the endpoint of the reaction. The concentration of hydrogen ions required to reach the endpoint is equivalent to the concentration of nitrogen that was in the original food. The following equation can be used to determine the nitrogen concentration of a sample that weighs milligrams using A into M, that is molar hydrochloric acid solution for the titration. That is percentage N equal to X moles divided by 1000 centimeter cube into gamma S minus gamma S centimeter cube divided by mg into 14 grams per divided by moles into 100, where the Vs and Vb are the titration volumes of the sample and blanks, and 14 grams is the molar weight of nitrogen. A blank sample is usually run at a same time the material being analyzed to take into the account any residual nitrogen which may be in the reagent used to carry out the analysis. Once the nitrogen content has been determined, it is converted to a protein content using the appropriate conversion factor, that is percentage protein equal to F into percentage N. Advantages and disadvantages. Advantages. The Kiljoule method is widely used internationally and is still the standard method for comparison against all other methods. Its universality, high precision and good reproducibility have made it the major method for the estimation of protein in food. The disadvantage is it does not give a measure of the true protein, since all nitrogen in foods is not in the form of protein. Different proteins need different correction factors because they have different amino acid sequence. The use of concentrated sulfuric acid at high temperature poses a considerable hazard, as does the use of some of the possible catalysts. The technique is time consuming to carry out. Enhanced Dumas method. Recently, an automated instrumental technique has been developed which is capable of rapidly measuring the protein concentration of food samples. This technique is based on the method first described by a scientist called Dumas over a century and a half ago. It is beginning to compete with the Kiljoule method as the standard method of analysis for proteins for some foodstuffs due to its rapidness. The general principle. A sample of known mass is combust in a high temperature, about 900 degrees centigrade, chamber in the presence of oxygen. This leads to the release of carbon dioxide, water and nitrogen. The carbon dioxide and water are removed by passing the gases over special columns that absorbs them. The nitrogen content is then measured by passing the remaining gases through a column that has a thermal conductivity detector at the end. The column helps separate the nitrogen from the any residual carbon dioxide and water that may have remained in a gas stream. The instrument is calibrated by analyzing the material that is pure and has a known nitrogen concentration, such as EDTA, that is equal to 9.59% of N. Thus, the signal from the thermal conductivity detector can be converted into nitrogen content. As with the Kiljoule method, it is necessary to convert the concentration of nitrogen in the sample to be protein content using suitable conversion factor, which depends on the precise amino acid sequence of the protein. The advantage of this technique is it is much faster than the Kiljoule method, under 4 minutes per measurement, compared to 1 to 2 hours of Kiljoule method. It does not need toxic chemicals or catalyst. Many samples can be measured automatically. It is easy to use. The disadvantage is high initial cost. It does not give you measures of the true protein, since all nitrogen in foods is not in the form of protein. Different proteins need different concentration factors or correction factors. because they have different amino acid sequence. The small sample size makes it difficult to obtain a representative, that is reliable sample list. Methods using UV visible spectroscopy. A number of methods have been devised to measure protein concentration, which are based on UV visible spectroscopy. These methods use either the natural ability of proteins to absorb or scatter light in the UV visible region of the electromagnetic spectrum, or they chemically or physically modified proteins to make them absorb light in this region. The basic principle behind each of these tests is similar. First of all, a calibration curve of absorbance or turbidity versus protein concentration is prepared using a series of protein solution known as concentrations. The absorbance or turbidity or the solution being analyzed is then measured at the same wavelength and its protein concentration determined from the calibration curve. The main difference between the test or the chemical groups which are responsible for the absorption of scattering of radiation, example peptide bonds, aromatic side chain groups, basic groups and aggregated proteins. A number of the most commonly used UV visible methods for determining the protein content of foods are highlighted, that is principle. Direct measurement of at 280 degree nanometer. 280 nanometer. Tryptophan and tyrosine absorb ultraviolet light strongly at 280 nanometer. The tryptophan and tyrosine content of many proteins remains fairly constant, and so the absorbance of protein solutions at 280 nanometer can be used to determine their concentration. The advantages of this method are that the procedure is simple to carry out. It is non-destructive and no special reagents are required. The major advantage is that nucleic acids also absorb strongly at 280 nanometer and could therefore interfere with the measurement of the protein if they are present in sufficient concentration. Even so, methods have been developed to overcome this problem, example by measuring the absorbance at two different wavelengths. Next is Biuret method. A violet purplish color is produced when cupric ions, that is Cu2+, interact with peptide bonds under alkaline conditions. The Biuret reagent, which contains all the chemicals required to carry out the analysis, can be purchased commercially. It is mixed with a protein solution and then allowed to stand for 15 to 30 minutes before the absorbance is read at 540 nanometer. The major advantage of this technique is that there is no interference from materials that absorb at lower wavelengths, and the technique is less sensitive to protein type because it utilizes absorption involving peptide bonds that are common to all proteins rather than specific side chain groups. However, it has relatively low sensitivity compared to other UV visible methods. The next method is Lowry method. The Lowry method combines the Biuret reagent with another reagent called choline Shea-Coltue, that is phenol reagent, which reacts with tyrosine and tryptophan residues in proteins. This gives a bluish color which can be read somewhere between 500 to 750 nanometer depending on the sensitivity required. There is a small peak around 500 nanometer that can be used to determine high protein concentration and large peak around 750 nanometer that can be used to determine low protein concentrations. This method is more sensitive to low concentration of proteins than the Biuret method. Next method is dye binding method. A known excess of nitrally charged, that is anionic dye, is added to the protein solution whose pH is adjusted so that the proteins are positively charged, the isoelectric point. The proteins form an insoluble complex with the dye because of the electrostatic attraction between the molecules, but the unbound dye remains soluble. The ionic dye binds to cationic groups of the basic amino acid residues, histidine, arginine and lysine, and to free amino terminal groups. The amount of unbound dye remaining in the solution after the insoluble protein dye complex has been removed by centrifugation is determined by measuring its absorbance. The amount of protein present in the original solution is proportional to the amount of dye that bound to it, that is dye bound equal to dye initial minus dye free. The next method is turbimetric method. Protein molecules which are normally soluble in solution can be made to precipitate by the addition of certain chemicals. Example, trichloroacetic acid. The protein precipitation causes the solution become turbid. Thus, the concentration of protein can be determined using measures the degree of turbidity. The advantage of this technique is UV visible techniques are fairly rapid and simple to carry out and are sensitive to low concentration of proteins. The disadvantage of specific UV visible spectrometry technique is that for most UV visible technique is necessary to use dilute and transparent solution which contains no contaminating substances, which absorbs a scatter light at the same wavelength as a protein being analyzed. The needed for transparent solutions means that most foods must undergo significant amounts of sample preparation before they can be analyzed. Example, homogenization, solvent extraction, centrifugation, filtration, which can be time consuming and laborious and be laborious also. In addition, it is sometimes difficult to quantitatively extract proteins from the certain types of foods, especially after they have been processed so that the proteins become aggregated or covalently bound with other substances. In addition, the absorbance depends on the type of protein analyzed. The different proteins have different amino acid sequence.